Check 1, 2. Check 1, 2. 2. 1, 2. Check 1, 2. That's me. 1, 2. This should be the Zoom coming through here.
So what's going into the Zoom then? There's nothing going into it.
No, this should be picking it up off of here.
But where's
when
it
I
going?
pick in the microphone here.
There's no input to it. There's just somebody has something plugged into the output monitor.
Oh, the output, yeah, the Zoom should be coming through here. You heard me on the Zoom?
On
Yeah.
the YouTube.
Yeah, so it's the speaker out of here,
Okay.
right, going into here. So whatever.
Nothing is going into that. Nothing is.
But there should this should be coming.
USB input.
That should be coming from. That should be coming from.
So that's just picking up the USB signal.
Yeah.
Okay.
That should just be coming as from one of these things here. Speaker 4. Let's try this. Speaker 4. It's which one of these? Connect speaker 5. Have them speak again.
Insoo, do you mind talking?
Testing, 1, 2, 3, testing. 1, 2, 3. Testing, 1, 2, 3.
All right. Okay, go ahead, Insoo.
Hello and welcome to the Biotechnology and Society webinar series. We apologize for the technical difficulties. I'm your host, Insoo Hyun. I'm the director of the Center for Life Sciences and Public Learning at the Museum of Science in Boston. The purpose of this webinar series is to bring the audience together with leading researchers in the area of biotechnology and to answer your questions that you may have about where the future of a particular field in the biotechnology area is heading. This is a series sponsored by the Center for Bioethics at Harvard Medical School and the Museum of Science here in Boston. So the topic for today is Parkinson's disease and cell replacement therapies. If you have questions you'd like to ask our speaker, you can go to slido.com, #MOS webinar, and submit your questions there. We'll get to as many of those as we can throughout the webinar. So it's my pleasure to welcome today's guest, Dr. Malin Parmar. She is professor and research team manager in developmental and regenerative neurobiology at Lund University in Sweden. Lund University has been a world leader in the area of Parkinson's disease and cell replacement therapies, and we're really honored to have Dr. Parmar here to talk with us about the past, present, and future of this area. She's joining us from Lund in Sweden. Welcome, Malin. Great to have you.
Thank you so much, Insoo. Thank you.
Yeah, it's wonderful. So I think in order to get everybody on the same page about what it is we're going to talk about, I'd love for you to just tell us a little bit about Parkinson's disease and also what traditionally people have been doing to try to treat this condition medically. You know, what are the standard things that people have done? And then maybe very soon thereafter, just talk about what makes your approach different.
Okay, so Parkinson's disease is one of the diseases called neurodegenerative diseases. And neurodegenerative means that there's neurons in the brain that die. So Parkinson's disease is the second most common of these diseases, whereas Alzheimer's disease is the most common. But they affect your body and your mind very differently because Alzheimer's is mostly about memory and cognition, whereas Parkinson is more or less a motor disease, although there are other aspects to it. So even though it is one of the most common diseases, we actually don't know much about how the disease arises or develop, but there's a few things we know about the disease, and some of them are important for targeting them for cell-based therapy. So I was wondering if the tech team can put on my first slide. So what you see here to the left is the brainstem of an individual that died without Parkinson's disease. And you can see in the little red box, there's a black area, and those are the dopamine neurons in the midbrain. And these dopamine neurons appear naturally pigmented, so you can see them with your bare eye. And if you look at the same brain region and the image below, you can see that these neurons are almost gone. And this is what happens in all patients with Parkinson's disease, is that these specific type of cells, the dopamine neurons in the midbrain, degenerate over many, many decades. So these neurons sit deep in the brainstem, but what they do is they send their projections up to the forebrain, and there they release dopamine, and dopamine is needed to control our movements. So when we lose these dopamine neurons, that leads to the motor symptoms of the disease, and it can be bradykinesia, rigidity, tremor, and defective gait. So it slows your movements and make it hard to control them. And this is because we have too little dopamine in the brain. And the most common treatment that is used today is then to supply this dopamine by oral medication, and L-Dopa is the most common form, but there's others. So what these do is they elevate the dopamine levels in the brain, and that makes it easier to control the movements. However, over time, it's really hard to control the correct dose of these dopamine medications, and they also lead to severe side effects and problems. So cell replacement therapy is an alternative to this, where we hope that we can replace the dopamine by replacing the neurons themselves. And this will give us a targeted and physiological release of the dopamine, meaning that we will get the same good effect as we get with the medication, but always with the right dose at the right time and without the side effects. So this has actually been trialed at the center in Lund where I work, where my colleagues Olle Lindvall and Anders Björklund and their teams developed cell-based therapies, taking cells from fetal tissue and transplanting them into the brain of Parkinson's disease patients.
I was wondering, people may have heard about deep brain stimulation for Parkinson's disease. Is that an effective therapy or intervention, and does that last for the lifetime of the patient? Tell me a little bit about what you know about deep brain stimulation.
So it can be a very effective intervention. It is used mostly towards tremor, for example, but also for other indications in the disease. And some, when the patients go into the complication phase where they don't get good effects of their drugs, you can use deep brain stimulation. It's a little bit different because what you do is you implant electrodes into the brain, and these electrodes either stimulate or silence different neural circuits. So you thereby modulating what happens in the brain, but it doesn't replace or reconstruct the same way as a cell therapy would do.
Now, the cell replacement strategy is meant to recover cells that are lost in the patient, the dopaminergic cells. Why do the dopaminergic cells in these patients tend to disappear? Like, what triggers or what causes that?
So this is one of the problems we actually don't know. In about 10% of the patients, there's a genetic component. So you inherit a gene that makes you more prone to develop Parkinson's disease. But in 90% of the cases, there's no clear reason for this. There are some indications that you can get it from pesticides or other environmental factors, but for the most part, we don't know why this disease arises. And this also makes it very hard to develop a treatment because we don't know how it starts. Now, cell therapy isn't a cure. The disease continues in the brain, but because we put in new healthy cells, the symptoms of the disease is mediated.
Yeah, so you are a research team manager at Lund University. Can you tell us a bit about the incredible history of this work at Lund University? What made Lund University sort of, one of the world's best centers for this type of research?
Yeah, so this is a fantastic institute to work in for this type of work. It was here during the 1980s and 1990s so at this time one thought that it wasn't possible to repair in the brain, but my colleagues Olle Lindvall and Anders Björklund really developed ways to transplant cells into patient brains and did the first pioneering cell therapy treatments for Parkinson's disease. And these patients have then, so there's patients operated in the 1980s and 1990s in Sweden, and then there's been a couple of hundred worldwide. But it all started here at the center where I work, and we've also been able to follow these patients for a very long time and to study the effects of the transplant over decades, and also to study their brains post-mortem.
So for that earlier work, what were some of the outcomes, and how long did apparent benefits of the transplant last? Could you tell us a little bit more about that trajectory, that timeline?
So there's a large variation in this. There's examples of patients that had a very good effect of their cell therapy or their transplant, and they could reduce or even get off all their dopamine medication, and this lasted for decades. So it is a one-time treatment. You get your cell therapy once, and that is supposed to last you for a lifetime. There's also patients that didn't get very much of an effect of their cell therapy. And there's also patients, especially in some of the US trials, that developed troublesome side effects, such as dyskinesias, which is over movement, that you move too much. So there's been a large variation in the patients receiving these fetal cell transplants.
So why do you think some patients did well with the transplant and others didn't? Do you have any idea about the difference?
Yeah, there's a number of explanations for this. So there's a number of differences in the trial. So the one thing is, if you look at the dose of cells, there's been everything between 1 fetal VM per brain to 6 or 7 per brain. So the dose has been very different. Another thing is that the immune suppression, because you need to immune suppress the patients for, we think, at least a year to get the transplant to stay and not be rejected. Some of these trials did not use immune suppression. Some used it for 6 months, and some used it for a year or longer. So there's fundamental differences like this, or also the way you implanted the cells.
Yeah.
The other thing is that what these trials show, that some patients are probably more likely to benefit from their transplant than other patients, and that has to do with age and the stage of the disease, etc.
Okay, so let me back up a little bit. So you were talking about the differences between the approach taken in Sweden versus the approach taken by investigators in the United States, the so-called NIH-funded PD trials. So tell me a little bit more about those differences. I think that's something I would really like to highlight. So there were some important differences. You're talking about immune suppressive therapies on the one hand, not on the other. So it's kind of like go down the column of what you did that's different from the US approach, and just to clarify that a little bit further.
So the trials, and it wasn't me, it was my colleagues that did it. Of course, I don't want to take credit for their work, but the Swedish trials, as well as some other trials in Halifax and other centers in the US, have been what we call open label trials. So there's few patients, the patients know they received a transplant. The investigator might be blinded, but it's still an open label trial. And there's complications with this, of course, but these trials really showed that in some patients, you could get very long-term effect of your transplants, although you didn't get it in all the patients. When the NIH trials were conducted, there was 2 conducted in the US around the same time. And actually, that one of them failed, came out the week of my PhD dissertation. So I entered the field at the absolute low point in the field. And that was, of course, very, very disappointing to the whole field, that these trials didn't meet their primary endpoints. I think in the one trial, a lot can be explained by the lack of immune suppression, and also the operation technique that created very concentrated noodles of dopamine neurons. So you get both an uneven spread, and this is what there's a lot of these side effects, dyskinesias, in this trial, and then also the lack of immune suppression. But even in those NIH-funded trials, later reports have shown, and especially from one of the trials, that if you take the younger patients or earlier on after onset, or look at the effect a couple of years after the transplant, there has been subgroups of patients that benefit from the transplant. So I think with that data set, when it was reanalyzed, it was really scientists from all over the world that had conducted both NIH trials and the open label trials came together to see what is the similarities? What are the differences? Why do we see what we do? And this was actually the start of another fetal tissue trial called TransEuro. That was an EU-funded trial that was led by Roger Barker in Cambridge, where patients were transplanted in Cambridge and in Lund. And that trial, we finished all the transplants, but it's still ongoing in terms of analysis. And in that trial, we've tried to do the patient selection and standardize the tissue as much as possible. But a major complication when you want, so even though there are differences in the trial design that could explain some of the outcome, a fundamental problem is that when you use fetal VM tissue that you get from aborted embryos, that is that each patient get a different composition of cells. And there's no way that we can quality test these cells sufficiently before we use them. So a lot of this variation could also be due down to like random variations in the cell material. Now, when we're moving into the era of stem cells, we can generate freezers full of transplantable dopamine neurons. And now we can really start to do much more controlled trials where every patient can get the same cells in the same dose. So it's also been a difficulty to sort out where the differences are, because the one important factor, the cells, you can never control using fetal VM tissue.
Yeah, that's very interesting. Could you tell me more about the network you're currently involved with and the so-called modern approach to cell replacement therapies? There's been a lot that's been done between the initial transplants that Ole Lindvall and others have done in Sweden to where we are now. So tell me a little bit more about this network that's currently, in play and the approach you're taking with stem cells.
Yep. So it would be great if you put on my next slide then. Because here I show you both the history and the status of the field. So down in the bottom left corner, you see the fetal VM trial. So this is a tissue source from aborted embryos. And you see pictures of Ole Lindvall and Anders Björklund that developed this therapy. And Ole was a clinician in these trials. So he really led the whole transplantation field forward. That was using this fetal tissue material. And then there's a picture of Roger Barker in Cambridge that leads the TransEuro trial, which is what we call the modern day tissue trial or fetal VM trial. And then what has happened in recent years is that we've been working in a organization called G-Force PD. And in G-Force PD, we try to use stem cells instead of fetal VM tissue to generate the cells for transplantation. So stem cells come in different flavors, and even within G-Force, we use two different types of stem cells. They're similar, but not the same. So on the left, you see human embryonic stem cells that come from very early human embryos. So this is when surplus embryos from in vitro fertilizations. And these cells, if they're left in the embryo, they give rise to every cell type in your body. And when you take them out and culture them, they divide a lot. You can make an unlimited supply of these cells. And at the same time, you can differentiate them or generate from them any cell type in the body. And we and our colleagues have figured out how do we control these cells from a stem cell to a dopamine neuron for transplantation. And the first trial was initiated I think about a year and a half ago. And the development of the cells in the trial was led by Lauren Studer from Sloan Kettering in New York. Mm-hmm. On the other hand, in G-Force PD, we had Jun Takahashi, and that's the director of the Sierra Institute in Japan. And he used induced pluripotent stem cells, which are a similar type of stem cell in that they divide a lot and they can generate all the cell types in your body, but you get them from reprogramming skin cells into stem cells. And from these stem cells, he initiated the first trial actually about the year before the American team in patients with Parkinson's disease. And then me and my team just initiated our clinical trial. I did the first transplant in that clinical trial, and that's called STEM PD. And we use here human embryonic stem cells, the same as a New York trial. So the field is a super exciting time of development where we see the first stem cell trials being brought from the research bench all the way to clinical trials in patients. So very, very exciting at the moment.
So the transplants you've done recently, was that also open label? Is that like the patients?
All these have been open label. You can take the next slide for a quick wrap up of the STEM-PD trial, which is a trial that we do now. It's a single arm, first in human phase 1/2a multicenter dose escalation study. So it's an open label in a single arm, and we use an allogeneic stem cell product. So every patient will get the same cells, and they're not matched. So we will immune suppress the patients for a year. They will be implanted into the putamen of the Parkinson patients, and the trial involves 8 patients in total. The patients in the STEM-PD trial are actually drafted from the Transgender trial, so we can compare in the same patient cohorts grafting with fetal tissue and grafting with stem cells. And I think you can take this slide down now. But all these initial trials are what we call phase 1/2a, which are safety trials. And they're all open label, which I think is essential when you do very invasive surgeries. You use cells with high risk components to them. So we want to use few patients in open label trials. There's a lot of complications with double-blind placebo-controlled trials in neural grafting. And some of that became evident when the NIH-funded trials were done. And it's actually kind of interesting because we looked into, of course, we look at what happens after a phase 1/2a trial, and we want to do global trials and bring them all the way to biological medications. So there, in the US, the FDA regulates these type of trials there, and they're very pro double-blind trials, and they probably require them. In Japan, you're not allowed to do a double-blind placebo-controlled trial in a neural grafting because it's too high risk for the control or the placebo patients. And in Europe, they're much more restrictive than in the US, but it is allowed with the right arguments. So looking globally, there's not a consensus among the regulatory authorities how you do this. There's, of course, if you do a standard clinical trial, double-blind placebo-controlled trials are very, very good and very, very important. But when it comes to these type of invasive stem cell therapies, there's a lot of complications that come with doing these double-blind trials. The one is the risk that you expose the placebo patients. So they go through hours of surgery. They're put under general anesthesia for many hours, which is not without the risk. So, and this is what the European authorities look at the most, the risk versus the potential benefit. And because they're placebo, there's of course no benefit.
Sure.
The other, sorry.
Oh, it's actually, I think I want to just pause here and just define some of these terms for our audience who may not know.
Yeah.
So a double-blind study is one where the investigators and the human subjects don't know who is getting what, whether they're getting the experimental treatment or a placebo. So nobody knows, and then there's a way to discover later who got what. So that's double-blind. A placebo-controlled trial is where you have at least 2 arms. You have one group that's getting the experimental treatment, so the active ingredient, they're getting the cells. The other group will be getting something that's not the active ingredient. It might just be, an inert substance, a placebo. So the controversy here is whether or not these kinds of neurodegeneration, Parkinson's disease studies using cell replacement strategies should be done open label. We used that term earlier. Open labels. Everybody knows who's getting what. There's no masking. That could potentially, for some people, initiate a placebo type response. That's one concern about that kind of open label. If you think that you're getting something that could help you, that might cause a psychosomatic kind of, interesting dynamic where you actually do you get some kind of benefit, even if there's no active substance being provided for your body. So there's an interesting controversy globally about how best to go forward with this. Is it open label? Is it double blind? Should it be a control group or a placebo group or not? What's really fascinating to me about the placebo proposal for this kind of strategy is you really have to trick as well as you can the patient into not knowing whether they got the real thing or just an inert substance, which means in this case, actually doing a sham surgery, actually putting them under anesthesia, making a burr hole in the skull, and actually, in some cases, actually even injecting the needle with nothing in it, injecting the needle into the brain and out. You can imagine all the risk that poses without any possibility of benefit for the human subject. So I'm just kind of laying out a little bit of sort of the landscape of terms and what the controversies might be. Did I get that right, Malin?
Yeah, that's perfectly. And of course, if you do a normal drug trial, not biologics, it's usually oral medications. So you can take a pill, and you don't know if the pill is active or if it's a placebo pill. And that's easy to deliver. But when it comes to these invasive therapies, they're hard to deliver. You have a lot of issues relating to the safety of delivery and the risk for the patients. And then when it comes to grafting in particular, we know that we need immune suppression for the cells to survive. So what do you do with the placebo group?
Right.
Do you immune suppress patients that didn't get the graft? Because it's not risk-free to immune suppress patients either. And if you don't immune suppress them, you don't keep the double blind, because it's easy to tell for the clinicians and their treating physicians if these patients are immune suppressed or not. So then there's a lot of these questions that comes in. I think in a way, we've had a lot of discussions with this in our global networks, and we've had discussions with the regulatory authority. And so one kind of compromise could be that you do 2 different doses of the cells, and the patients don't know, or the clinicians don't know which dose of cells they received, instead of doing the classic dose escalation that we do in these early PD trials, where we transplant. Now in the STEM PD trial, we transplant 4 patients, assess the effect on the cell survival and the safety of that. And then we decide if we increase a dose, and then we set an increased dose. So a way to get almost like a double-blind placebo control would be to mix these doses, and no one knows what dose is had until you break the code. Or in the open label trials, of course, we do what we can to remove. There's both what you talk about is a patient bias. They get the treatment that they think might be effective so they can feel better. We try to assess with unbiased measures that can measure the cell survival and the function. But the other thing that you shouldn't forget is also the investigator bias. Because, we've all worked in this field for decades. We want our therapies to work. And if we know what patients have got in the treatment, we might be a little bit optimistic when we do the ranking of the ratings. So in the trial designs we have going now, both for STEM PD and TransEuro, so all the patients are filmed, and they're ranked or rated by the same neurologists that are not the neurologists that conduct the trial, and there's controls and there's transplant patients, and we don't know. So the clinician that decides the effect of the therapy do not know if it's a transplant patient or not. So there's in between mixes you can do, but it's an interesting and very complicated question, because there's no good, straight, and easy answer.
Right. We've had some questions come in through Slido, so please, audience members, if you have further questions, submit them. We'll get to those questions very soon. Well, and I was wondering about some further challenges in thinking through, the trial. You said that this was initially going to be just a safety study, right? So you've done transplantations and safety. Will those participants get a chance, maybe if it's proven to be safe in this round, will they get a chance later to get a higher dose, or are they kind of done with trial participation? You would have to go to a different group of patients for the next stage of research.
Yeah, you would have to go to a different group of patients, because you couldn't repeat transplant the same patients. But the dose we start with here, we're really helped by the fetal VM work and all the work by Ulle Lindvall and his colleagues earlier on, because we know approximately how many dopamine neurons we need to get the clinical effect in patients. So when we select the minimal dose, this is also a little bit different than a standard drug trial. There you do a sub-threshold of what you think is effective. But our minimal dose is the dose that we think is going to be effective in the patients.
So what kind of things are you looking for the safety trial? Like what are you hoping to discover?
So in the safety trial, it's the first human dose trial. So you look at all expected and unexpected, what we call adverse events. So anything that could potentially happen that's related to the cells or the surgery. And that's the primary outcome. But in secondary outcomes, we of course look for do the cells survive? Do they produce dopamine? And do they mediate functional effects in the patient? So they reduce her Parkinson's symptoms. So this is measured in different ways, both by brain imaging, where we can hopefully see, because we haven't gotten to that point yet, but where we hope to see cell survival and production of dopamine of the neurons. And then we, of course, also do a lot of clinical rating and self-assessments in the patients to see if we can pick up improvements in movements or cognition or the reduction in their dopamine medication that they take, etc. So these are all what we call secondary endpoints. So a clinical trial always have one primary endpoint, and in this case it's safety, but that doesn't mean that we look only at safety. We also look at effectiveness, because there's no point to have a safe therapy if it's not also effective.
Right. Now, Parkinson's disease is a degenerative disease. It's very gradual. What stage of the disease are you using for the patients that are in this very first step? Are they early? Are they early on in the process of Parkinson's disease? Are they in the middle, toward the end? Where are they in that trajectory?
So we think that the best stage to deliver the therapy, if we knew it was safe and effective, so say that we fast forward 8 or 10 years, it would be right after diagnosis. So the first line treatment, you get your diagnosis, you get your cell therapy. But these are patients that have many years of good dopamine medication left. They have a lot of treatment options. So until we know it's a viable and competitive therapy, that's not the patient population that we start in. In the US and the Japanese trial is late stage Parkinson's disease, but also these patients are almost at the stage where they have very little benefits from the transplant. So the European trial, STEM-PD, is in moderately advanced patients, where they run into complications with their normal oral medications, but they're still not at the late stages of the disease. So these are the patient population that we elected for our clinical trial.
I want to get back to the question of the sham surgeries. There's some who would argue that that's very valuable to know what effects some of the procedures may have on the at least maybe the nearer term outcome of the patient. So some people may wonder whether or not just even putting the needle into the brain without anything in it, but just the needle in that region, taking it out, has some kind of effect. And would it be, how would you sort of separate out that noise or that kind of, confounding factor from the effect that the cells are actually having in your study?
Well, there's none of those trials going on right now, but I would assume that you actually don't inject a needle. I think that you make a burr hole, but you never actually puncture into the brain. And this is very important because the brain is what we call immune privileged, or it's protected from the rest of the body. So we have the immune system that comes with our blood system, and that doesn't really have access to the brain. But if you so there's something called the blood-brain barrier. So there's a barrier that protects the brain from the rest of the body. Once you do a surgery, you open up this blood-brain barrier and you can have infiltration of the immune system. So this is not something that you want to expose anyone for, but you would just the general anesthesia itself is not risk-free. Another thing to take into consideration in the discussion of placebo-controlled studies is that when you do a cell therapy, it takes at least 6 to 12 months before you see an effect. And in clinical trials, we know that the placebo effect usually comes very early after treatment.
Interesting. Yeah.
So because we transplant the cells when they're immature, they need to mature into functional dopamine neurons. They need to send out their projections to the brain, and they need to start producing and releasing dopamine. So in the fetal VM transplants, often it took at least 12 months before you could see the effect, and then the effect steadily increased over the next subsequent years. So usually that's not how placebo effects would present themselves in the patient. So this is also an argument against the need or that lowers the need for placebo-controlled trials in this type of invasive surgeries.
Mhm. Very interesting. So I just want to just recap a little bit for the audience, some really important differences between past approaches for cell replacement therapies and the current one. So in the past, you said, Malin, that you had to actually put together several samples from fetal tissue. So what happens is a woman elects to have an abortion, and then once a decision has been made, then the researchers ask if they could use some of the biomaterials from that abortion for the transplant therapy. So this would be taking the dopaminergic neurons from the sample and transplanting them, but just one sample doesn't have enough for an adult. So you have to actually put together several from different sources. And so it's really hard to kind of control in an experiment, right? I mean, possible to control the uniformity. But now what your approach is going to be able to do is grow from a cell line, grow from a stem cell line, the exact type of cells that you would have used in the past from the aborted sample. You can create the exact type of cell, do quality control, get the exact same profile across the board. You're putting the same product into every subject. So it's like a drug. You can kind of, measure and keep the uniformity. So that's a huge advantage, right? The ability to make the cell type that you need from a cell line and then conduct your experiment. That also could mean that in the future, if this does prove to be safe and effective for patients with Parkinson's, that you can actually manufacture the product for dosing many other people in the future.
So it's both an availability, because in the TransEuro trial, which is a fetal tissue trial that we conducted in the last couple of years, we had to cancel over 100 surgeries because of lack of tissue. Because it's not, we know from the earlier trials, so you need at least tissue from 3 aborted fetuses. And these need to be collected within 5 days. And it's very hard to actually achieve that. And many times the surgeries had to be canceled. And this is also a difference, if you go back to the difference between the NIH trials and the earlier open label trials, is that in the NIH trials, to get enough tissue, they often kept them in the fridge for weeks. And we don't know what that does to the viability of the tissue. And the other, when there's such limited amount of tissue to use, there's nothing we can do to quality test the cells before we use them, because then we don't have enough cells to use in the patients. When we look about the stem cells, we can produce unlimited numbers of cells. We have 300 doses in the freezer. We can use those exact same cells in the safety and efficacy studies in animals before we go into patients. So we can do much more available therapy, but also much better controlled therapy. And for example, the dyskinesias, the side effects are thought to be due to contaminating neuronal population, the serotonergic neurons. And those we can completely exclude when we do a stem cell differentiation, but we can never exclude it when we do dissection from fetal tissue.
Yeah. So one of the key decisions that any team has to make is when they're going to conclude their study and report on the results. And so, one might say, well, how long after the transplant into the patient should we say that their trial has ended and we're going to, take our measurements and report the results? Is it 1 year? Is it 2 years? The Swedish experience shows that it could be many years before you get a really sizable effect and benefit for the patient. And in fact, even the NIH studies suggest that in the initial round of reporting, there was really nothing, that astonishing or beneficial for the patients. But when they looked a little bit further out, a few years out beyond that initial reporting point, there did seem to be some benefit for some of the people who initially were thought to have not benefited. So how do you decide how long you want to keep the people in the study and report on your results? Like, when do you think should be the conclusion of the trial? Is it 2 years, 3 years, longer than that? How do you know how far into the future, to keep going, to know whether or not these are helpful therapies during the lifetime of the patient?
Yeah. Now, this is a super relevant question, because it's a very difficult to develop therapies that take so long before you get an effect, makes the trials very long. Now, with the stem cell trial, it's a safety trial. And the primary endpoint is safety, and that we do after 1 year. So we can conclude that after 1 year, although we continue to look at the secondary endpoints after 2 or 3 years, which is efficacy endpoints. When we look at the Transuro trial, which is the fetal tissue trial that's currently ongoing, their efficacy, there we want to look at the efficacy of the transplant, and the primary endpoint is 3 years. And that's enormously long when you conduct clinical trials.
That is very long.
Yeah.
Ole Lundvall has told me that this is sometimes, he refers to it as lifetime research. I mean, you have to sort of look at the lifetime of the person. When you eventually go to a clinically, what you think might be a clinically significant dose, do you have any plans for, because I know in the past with the fetal transplants, there have actually even been cases where you did autopsy on the patients who eventually passed away and found that there were actually, the cells were still there. They were doing their job. Is there a plan with the human subjects in your upcoming trials to maybe get consent for something like, in the case of eventual death, you'll have an autopsy, you'll be able to kind of examine how well the cells, stood in the body?
So I just want to highlight from what you said that the dose that we start with, we think is clinically effective. So we chosen the lowest clinically effective dose as our low dose. When it comes to looking at the brain's postmortem, it's extremely valuable to do so. We're not asking for brain donations when we ask for consent to be part of the clinical trial, but we will implement this later. And this is also a benefit of working in Lund or Sweden. It's a small city. In Sweden, we have this person number, so we can keep track of everyone in the healthcare system, and we can find these patients 20 or 30 years later. But it's a fantastic. So the work that Ulla did in the 80s and 90s, when those brains finally came to postmortem, many years later, we could learn a lot about the transplants and what happens in there and how well they survive. So of course this is a valuable resource, but that's, if the patients are maybe 60 years old when they get their transplant, we're hoping it will be 25 to 35 years until their brains come to postmortem. So it's a long-term effort.
Yeah, I want to take up a question that's come in through Slido. And the question is, how do you foresee equal access to these types of therapies playing out in the future, right? So in the best case scenario that this works and is effective, I think there are general public concerns about access to these therapies for patients in need. Do you think this is going to be something very expensive for patients, for the healthcare system? How do people who need this therapy, have you thought about how they might access it?
So that's important and relevant questions. And so stem cell, like the production of the stem cells in the first clinical trials are very expensive. But over time, I don't think that these therapies have to be enormously expensive. And you also have to consider, depending on what health system you're in, but it's a one-time treatment instead of medications, other invasive therapies. The patients, we hope, can go back to work so they can contribute to society. You delay the time until they need care home care and these type of things. So from a societal health economic aspect, it might be actually cheaper than it is with the standard medication therapies once these are developed therapies. But there is a cost to them at the moment that I hope can be reduced. The other important question is, of course, how specialized is this? But the surgery itself is the same as more or less the same as when you do the DBS therapies that you talked about earlier. So any DBS surgeon could easily also perform cell transplants. So it could be available at the same centers. Another question when it comes to accessibility is whether you use human embryonic stem cells that come from surplus embryos, human embryos, or if you use iPS cells, because there are countries today where it's not possible to deliver therapies based on embryonic stem cells, for example.
So you said that in the US, the regulatory bodies really like double-blind, placebo-controlled trials, and the Swedish approach, and I think the Japanese approach will not take that route. If the innovations and the breakthroughs happen in Sweden and Japan, can those therapies be imported into the US, even though they haven't kind of gone through the FDA's preferred, double-blind system?
Well, I think the trial design, and this is a little bit looking into the future, but you do a global trial, and then the placebo patients would be in US and some in Europe, and there would be no placebo patients in Japan, but the transplant patients would be there, so that you would go for a global compliance from the start, because these trials are too expensive to conduct in at each region. So,
Sure.
So I think that can be mediated.
I'm going to ask you a question, my last question that I think a lot of patients ask you, and that is, what is the expected timeline if things work out in these trials of when people could see therapies, cell replacement therapies in the clinic?
Yeah, so when it becomes approved therapy, I think we're at least 10 years from that now. And part of that is because the trials need to go through the phase 2 and 3. You need to make sure that what we implement is safe and that it is effective. And the reason it takes a long time for this type of therapy is exactly what we discussed with the endpoints for the trials. So if the endpoint is 1, 2, or 3 years, it takes a long time to go through the different phases of the clinical trial. So it is, optimistically 7 or 8 years maybe, but realistically around 10 years time before I think this can be a market approved drug.
Well, I think we're in a very exciting time. The major innovation in my mind is being able to pivot away from using fetal tissues and being able to grow the cells that you need for the transplant from a cell line. I think that opens up many, many other opportunities. So thank you so much, Malin, for sharing all this with us today. I really appreciate the update, and best of luck to you and your colleagues on your work going forward. So that concludes our webinar for today. I thank you and your expertise for joining us today. I also want to thank the Center for Bioethics at Harvard Medical School and the Museum of Science for co-sponsoring this event. I want to thank you, our audience, for joining us. I hope you learned, where things stand right now with Parkinson's disease research and cell replacement therapies. My name is Insoo Hyun. I'm your host. Thank you for joining us today. Have a great rest of your day.
Thank you.